Hi everyone. In this video, we will explore EasyCellType, another R-based tool for marker-based cell type annotation. Basically, it takes your gene and it compares against the marker database, and it uses two statistical tests. Okay, one is the gene set enrichment analysis GSEA based enrichment scores, or the alternate is the it gives Fisher exact test based scoring it gives. Okay, and yeah, and the results generally presented in two way, like hard classification and soft classification. Hard means it will gives the most possible cell type for each cluster, so that means it's a hard bargain. That means it will give you the best one. Okay, but soft will give several top possibilities. So depending on when the confidence value what you get, right, you can go for try with hard as well as soft and see how the assignment looks like. For example, that's what here you will see that for cluster 1 CD4, whereas cluster 2 B cell, but here you have multiple types, you will get soft classification. Okay, and the logic is like it takes the expression scores and it against databases, and based on the enrichment scores or Fisher exact contingency binary classification it does, and based on that it tries to uh, generate the cell type. Okay, so yeah, so these are the things I have. We'll see. And so this is the easy cell type website you can search. And yeah, so they have provided nice Signy app as well. Okay, so you can just annotate your data here. So I will just upload an example data CSV file. Okay, and these are the entries ID in symbol. So here we can choose which database we want against. So by default in like within the easy cell type package itself, we have these three databases are already loaded, like CellMarker, PangelDB, and ClusterMole. ClusterMole itself has 7 database, right? So that way it is more comprehensive, you can say. Okay, so you can choose what database you want and then choose a species, and you can choose a tissue. Let's say in this case we can, okay, we can do blood, maybe immune. Oh, blood, let's say blood vessel, something peripheral blood. Just I'm just select, and now you can choose which test. Fisher exact test is more faster, and GSEA is a little slow, but it will run. Okay, so if you just run, right, yeah, it gives the same plot. So basically what these two plots are very important. So this the x-axis, you see the cluster 0 1 2 3 4 5 6 clusters, and y-axis are the cell types, and the bubble indicates like the I think the score what we get, uh, and the p-value, probably that is what it is getting used. And so it says that 0 is more of towards T cell, it is going right. Similarly, cluster 1 it is going towards helper T cells, that side. And cluster 2 is going more towards B cell, it will go towards the B cell, right? Okay, I don't see that B cell, right? Okay. But here, let's go by one. So here for every cluster 0, you will see the confidence. Okay, what's the score I was thinking? This is the p-value minus log 10 p-value, So higher is the value means more is the quality confidence. So you can say that way. And so here you see there is very much similar across all this one. Cluster 1, like everything is equivalent because T cell. Cluster 2, we got only, what is only T cell it is showing. It should have shown the B cell as well, right? I think this y-axis is fixed, top 5, and it is showing for these 5 cells, and what is the how the marker goes for each clusters, that way. Okay, so yeah, but that is a simple way you can upload your data and you can compare against, let's say, cell marker only. But let's try GSEA. Okay, as I said, GSEA might take some time to do. Yeah, it's finished. Yeah, so cluster number 2, see B cell is high, and but again it predicted natural killer T cell. Okay. Yeah. So you'll have to verify, like enrichment versus Fisher exact test, what you want to use. Just thinking any tutorial is given. Yeah, it's not loading, it takes time. Yeah. So they have, as I said, they have given these bioconductor packages, right? So we need to install that and we can follow the same markers here. But only thing, like, we need gene ID. Okay, it doesn't recognize symbol. That's what we need this org, this package to convert that. And yeah, this is the one function where you are passing the input and what database you want, what species and what tissue you want to filter and cut off. So that way we will be able to do that. Okay. So yeah, so I have in the code backend the same thing I have tried. Let's see. So we will load this all these databases, and now just process our Surat pipeline to this one top markers. Okay, but here we are just using the gene, the cluster and average fold change, and now we will be converting that into a symbol, gene symbol to gene ID conversion we will do. Okay, so let's see, it is running now. So this is the data we have. This is the gene, this is the cluster, and the score is this one. Okay, expression. Perfect. So now let's do the we use this map ID function to map the gene symbol to this one, and we are doing it. So here just we are seeing how many percentage we could map from gene symbol to gene ID. So 93% we could map it. Okay, so these are the unmapped one, put it here, and just some sample name, gene symbol names which are unmapped one. Okay, so now what we are doing here is like any remove those unmapped genes. We have filtered it. So now this is the final data what we need to pass it. Okay, so now just let's explore what databases it has. So three database, right? CellMarker, PangloDB, and this one, right? So we, by using this data function, we can load that. And yep, it has, see, mouse, it has human, 159 data is there, maybe. So in case of PangloDB, we have the data is there for mouse as well as for human. Okay, and what are the different organs it covered, that it shows for each of the database. So now we have the third one, cluster mode. Okay, which is third one, and for human it is around 80, and mouse around 33 organs data set are there. Okay, so now for PBMC, what we are using, we need to blood, peripheral blood, blood vessels, anything blood is there that we can take. Okay, so now first step what we will do, we will annotate it using GSEA, then we will annotate using the second approach, that is Fisher exact test. Okay, and we can choose what database we want. So first we will do GSEA with CellMarker database. So here you give that out input, the database select CellMarker, species human, and these are the tissue we were filtering, and p value cutoff you can reduce to 0.05 as well, not a problem. Okay, so I'm just giving higher value. So this one will take some time, I think. Okay, so let me pause my video, then we will continue. Okay, yeah, so it completed now. It took some time, 1 minute to 2 minutes it took. So now, so what we did, we run this function, we gave the input where we have the gene list, we choose the database and species, and these are the tissue against which we are calculating. And so we will see, like, we got around 9 this one. Okay, 9 clusters, right? Output is a list with results per cluster. Okay, just you see the results for the cluster 0. Okay, for first cluster, what it is, like it give all the rank wise. Okay, so the enrichment score is highest for this one, naive CD 8 plus T cell. Okay, and followed by the other enrichment, not highest actually. Yeah, so it ranks how I'm just thinking. Okay, it gives this q value, which is the significance wise. I think that is sorted with respect to this p adjusted or q fdr value, right? Probably that. Okay, so that means most significant one is on the top, not based on the enrichment score. Okay, so it is ranked as per the significance. So this is the first one. Okay, yeah, p adjust is bh bond correction adjusted p value, and core enrichment column what we see here, right? Okay, so these are the gene IDs. Okay, because it's gene symbol is not there, right? It uses the gene IDs. So the marker genes that is giving the enrichment. Okay, and if you want to see the data here, so we are the not the gene symbol, we are using the gene ID. So that's where in the enrichment you see that for this cluster 0, the most significant one is this one, and these are the marker genes that are enriched in this particular cluster. So like that. Similarly, you can also visualize the second one. Okay, so if you want to go to, let's go to third is the B cell, I think, right? B cell, cluster number 2 is the B cell, which we already know so far, and we can see that this is also predicting around a B dendritic cell. Okay, so that way the top one is the q value adjusted one. Okay, perfect. So now the second approach, let's do same tool, but only thing like we will be now using Fisher exact test. So what is Fisher exact test? It is basically 2 by 2. It generates a contingency table. So you have your gene, you have the marker gene, whether that gene is present there or not. So that way it will the in that cluster. So for each cluster to cell, for each cluster to cell type pair, build a 2 by 2 contingency table, and it tests whether the overlap is greater than by chance or not. Okay, uh, okay, and yep. So this is very fast. So here just we have to change the Fisher here, but rest everything remains same. Again, we are using CellMarker database only, database CellMarker, but we are comparing GSEA versus Fisher exact test. Okay, so we did that. So now you see this is the cell name and the p adjusted one and the enrichment score. Okay, so what enrichment we got, and yep. So now, so let's try one thing. Now in the next step we will compare against 3 database. Okay, so this way we are getting different results here. Okay, so what we will do now, okay, I forgot to do that here. I think which cluster goes here. So here in annotation GS we got this one. Annotation Fisher is yeah, I have to see, what is the cluster went, right? Oh, okay. Sorry, my bad. So this will be your 11. Okay. So what I showed here, right? I got confused. So what I showed here, this is the cluster 1 all cell type detected. Okay. And the top one is the monocyte. So as per this Fisher exact test, monocyte is the most probable cell type, and these are the enrichment marker. Okay, and if I want to know about the second one, so that is also monocyte, and if I go to third one, that is the B cell, most likely it will come. But see, it's everywhere monocyte is coming. I don't know how it will behave. Let's see. Okay, so that's what I went. Now let's say run against all the three database for comparison. Since it is very fast, right? So I choose Fisher actually, but I could have done with GSEA as well, but that's fine. So let's do one thing. Run the same against Panglao now. Okay, and store that here in this variable. So now we are running against cluster mole here now. Okay, so now I will have to extract the hard classification and the soft classification. Okay, so I just wrote a very small function here. So I'm getting hard levels from each of these three results from these three database. Okay, okay, so we are just, don't worry about the code, just to show you what I'm doing here. For every cluster, CellMarker gave this one, PangloDB gave this one, and as per cluster mole we got this one. Okay, and these are the adjusted value like that. Similarly for cluster 1, the how it is, for cluster 2, how we are getting the different information. Okay, from this approach. Okay, and if you go by soft level, so soft level means we will get like this, cluster 0 will get more than one, right? So three types, cell types we got, not just one. So hard means we'll always get only one. Okay, and so we have several built-in plots that we can explore. One is the dot plot and one is the bar plot. Okay, same information we will get here in a different ways. So here the dot plot and the bar plot. So this is the dot plot. We can compare between GSEA versus Fisher exact test. You will see that it's very different results what we are getting. For the cluster 0, we are getting this one, right? Bubble very high, CD T cell here, and here cluster 0 we are getting CD8. Same only we are getting actually, but different distribution you see. And cluster number 2, you see towards the down we are seeing B cell, B cell, B cell here, and cluster 1, 2, where is 2 gone here? Okay, I think it might have missed, I don't know. 2 is missing here. But Fisher exact test, it gave all the cluster, no, 0, 1, 10, 2. Between 10 to, okay. Yeah, maybe, it has missed that probably. So yeah, this kind of plot dot will give you this kind of plot easily. And there is something called bar plot that will give you the bar plot. On UI we saw, right, the same only you can generate over here. Okay. So the code is like plot GSA, and you can save this plot. Okay, so let me go back and zoom it here. I'm just thinking maybe 2 is there, but 2 is missing. So in GSEA 2 was missing. Probably it's not there. Okay. Enrichment. So in the bar plot you will get this PDF file. Okay. So we got a PDF file like this bar plot, right? So for every cluster 0, which cell got enriched. For cluster 1, how it is in same what we saw in UI, the same only we are getting nothing different here. But programmatically, if you want to do, then this is how you will going to do it. Okay. And yep. So this is something I tried actually. Let me see what is coming. Okay. I also don't remember now. This is the p-value matrix. Oh, p-value matrix confidence. Okay, this is an interesting one. So what I did here, we got the p-value, right? For every cluster, we got some p-value for a given cell type. So we get a matrix. You just do the heat map kind of thing. So now you see what it interprets. You can see that these two values is very high, right? So you can directly go there and that will say, okay, these are the T cell like this. And cluster number 2, we got our somewhere directly little light B cell. Cluster number 3, these are the enrichment. Cluster number 4, these two are the highest one. So based on that, p value, we can also get another information about the confidence. What else I had here? same information, but very differently. Now you see, this is the confidence p value. High means green color, low means like orange one. Cluster 6 is very low confident, and medium is okay, and the second is not significant at all. So that's what I think it missed in the Fisher exact test probably. Okay. So oh my god, what else I had? Yeah, this is what finally long format is the right again. Okay, this is the final summary what we did for 3 databases. CellMarker 0, naive T cell, bone marrow T cell, like that. Second one B cell, B cell, B cell, but confidence size is very low here. Okay, cell as per CellMarker. So like that also you can do the plot. Okay. So why we are doing, first of all, if you see, right, because there are so many databases, you cannot rely on one database to get the confidence. So that's why it's always better to try with multiple database and come to conclusion and see that. Okay. And yeah, so then finally you can assign with some of the levels, any one level, and we can do the final plot here. Okay. So this is the CellMarker based Fisher exact test, CellMarker, and EasyCellType annotation. Okay. So in short, right, when you want to use this tool, you can use their online UI portal to annotate it. But if you want to programmatically do it, so you can choose GSEA or Fisher exact test, and mostly you can go with the hard one to get the initial consensus, because you have 3 flavor of databases, right, to choose, and you will get different kinds of plot over here. Okay. So yep, so these are the 3 databases we are comparing here, and yeah, ultimately, so in short, so don't worry about this might look very complex, but so once you have the code, this is all documentation are there, right, all these tutorials are there, you can follow that, but try to understand main thing, like when we are doing it, right, we need to understand the biology. So because these databases will give you any different names, so we need to correctly uh, don't trust the output what these tools will get, because they are randomly comparing, right? So that's where 2-3 databases through 2-3 tools we need to do, and we can come to the final confidence, like, hey, all the tools are behaving this way and all the marker databases are giving same information. So I think this is what the right one. Okay. So with that, I will conclude here the marker based. So far we covered what we started with our own approach, one method. Then we went to go with the SCType. We saw that. Then cluster mole tool we saw, and last we saw the EasyCellType. So 4 tools we saw, 4 different approaches and multiple databases. How you are going to compare all these 3 to come to a conclusion that we saw. And that's where we will move to the next approach that people follow, that is the reference based single cell annotation. So basically we will take our cell data, expression data, UMAP projection data, we will put it into another data. Okay, so that's what we will be exploring. Some tools, the most popular one people use single R, which is actually correlation based, and Agimoth is another one which is projection based. And the third one is like training machine learning models based methods are there, like CellType based and SCA and AVI. So I will see, like, which one to choose. I have not tried this one, but yeah, mostly I have tried CellTypist. So these 3 I will cover. Okay. And so then I think we'll move to the next reference based this one. So yeah, I have some information. I've already compiled that. So yeah, lot of excitement stuff, and I hope this was useful. And yeah, I will see you in the next video. Thanks for supporting the channel and watching it, and do provide feedback, because this will help me to understand how you are watching it and gaining the content, and suggest if you want to learn something more. Okay, with that, let's stop. Thank you so much. I will see you in the next video.